Okay, so I hope you see my screen. Welcome everyone. Today is the second event of this year for the Ask the Expert series. Today's event is titled Let's Trick the Skin. So obviously it's on transdermal drug delivery. This event is organized by the CRS, the Young Scientist Committee. The YSC, or the Young Scientist Committee, is a committee of a diverse group of students and early career scientists in academia or industry that are committed to developing scientific and professional development workshops, as well as organizing networking events. First, I would like to introduce myself. My name is Hagar Labuta. I'm assistant professor at the College of Pharmacy at the University of Manitoba in Canada. So I will be the moderator of today's event. Before I start introducing our invited speakers, please keep your your mic muted all the time unless you are asked to or invited to ask your question, just to enjoy a really good quality of sound throughout the event. Also, please feel free to ask your questions even throughout the presentation and the discussion, and then I will be asking you to say your question, or even question according to your preference. So let's start with inviting or welcoming everyone today. So today we have really three speakers who are expert in this field. First, it's really our great pleasure to welcome Dr. Mark Prausnitz. Dr. Mark Prausnitz is a professor in chemical and biomolecular engineering at the Georgia Institute of Technology. His is in chemical engineering from Stanford University and from MIT. Dr. Prausnitz and his team carry out research on biophysical methods of drug delivery using microneedles, lasers, ionic liquids, and other microdevices for transdermal, ocular, and intracellular delivery of drugs and vaccines. He published more than 300 journal articles and has co-founded 7 startup companies, including Micron Biomedical and Clearside Biomedical. So we are happy having him today to talk more about the translation of these products. We are also happy to have today Dr. Sarah Hedtrich, assistant professor at UBC in Canada, with a professorship at the Berlin Institute of Health at the Charity in Berlin in Germany. Dr. Hedtrich obtained her PhD in pharmacology and toxicology from the Freie Universität Berlin in Germany, and postdoc trainings in Ludwig Maximilians University in Munich and Tufts University in Boston. She authored over 80 journal articles. Her research focuses on inflammatory and genetic diseases of the skin. She's also focusing on development of novel therapeutic approaches. And interestingly, she is also developing complex tissue engineering models and human-based organ models for the aim of valid and predictive test systems for preclinical and fundamental research. And we are looking forward to hear more about these models during this event. We're also happy to welcome our proud YSC member, Dr. Anna Malero, who is a full professor at the Faculty of Pharmacy at the University of Valencia. She studied pharmacy and did her PhD in the University of Valencia, followed by a postdoc at Zarla University in the team of Professor Klaus. Her team focuses on the design and characterization of advanced drug delivery system to optimize availability through the skin for systemic and topical applications. She's currently working on the application of ultra-flexible vesicles through the skin for the treatment of inflammatory diseases such as psoriasis and atopic dermatitis. So we're happy having you today, Dr. Melero. These are the three speakers who are joining. Before we start, I would like to draw your attention that the biggest annual meeting for drug delivery research is coming up in July in the beautiful city of Montreal. So we're having really a full board of speakers and really exciting events. The YSC are also organizing several events in this meeting. So if you haven't registered, I would really recommend that you register as soon as possible. So again, we're welcoming the speakers. And before we start the discussion, I was also a YSC member that will give us a short presentation on transdermal drug delivery and some challenges in clinical translation. Before Dr. Sara Cordero starts the presentation, I would just like to introduce her as well briefly. Dr. Sara Cordero is a senior lecturer in pharmaceutical sciences at the School of Pharmacy at De Montfort University in Leicester. She has a PharmD from the University of Porto in Portugal and PhD in drug research and development from the University of Santiago de Compostela. Hopefully I pronounced it correct. And she has a postdoc from Queen's University, Belfast. She is an expert in nanomedicine and microneedles for transdermal drug delivery and diagnostics. Her research group focuses on the development of drug and vaccine delivery systems that are easy to manufacture, scale up, and that are highly efficient and administered through non-invasive routes. And that's why she is really another excellent fit for our event today. And I invite Dr. Cordero to start the presentation.
Thank you so much, Hagar. It's really, really a delight for me to introduce the topic that will be discussed by this excellent group of friends, collaborators, mentors that gathered here. And I really feel I don't live up to the title of expert in microneedles when Dr. Prausnitz is in the room, but I'm trying to continue learning on the topic. So yeah, today we're going to talk about tricking the skin, and just to introduce a little bit for those who may not be so familiar with this route of drug delivery. Being the largest organ, obviously, the skin is a very convenient route of administration for patients. It importantly avoids the first pass metabolism of drugs, increasing the bioavailability of those drugs, and allows controlled drug release. And it can also avoid drug degradation by the GI tract conditions that usually are harsh for drugs, particularly for biological drugs, which are a lot of the new drugs coming up. So if you can, I don't know if I can control the presentation. No, I don't think so. Yeah, perfectly. So, however, most drugs cannot go through the stratum corneum, which is the outermost layer of the skin, and only a very restrictive elite of molecules can passively go through that barrier, which is generally molecules that have low molecular weight, some degree of lipophilicity, and are given at lower doses, such as less than 10 milligrams per day. So that means that we need better strategies and better approaches to go through the skin and to allow transdermal drug delivery. And there's a variety of approaches that have been developed in the last few years. I've just highlighted a few here. On the left side of the slide, we can see a laser-based needle-free injector that was recently certified as a medical device for the European Union, or by the European Union in 2020, and has been used to deliver some drugs through the skin through jet injection. Some nanosystems, particularly lipid-based nanosystems, of which Dr. Melero is the expert here, have been also developed to facilitate transdermal drug delivery. And then obviously, I'm fond of our very dear microneedles, of which Dr. Prausnitz results there show that very easy to manage patch that his group has developed for measles and rubella vaccines and other vaccines as well. However, if we can go to the next slide. And if we look at the market trends in transdermal delivery, you can also see that the market is growing in this area, and it's majorly driven by an increasing prevalence of chronic diseases. Particularly cardiovascular sector has been the fastest growing segment, with some transdermal patches being developed for the treatment of angina pectoralis, for example. And there's obviously an increasing inclination of both patients and physicians towards pain-free drug delivery. So there's also a high demand for self-administration of drugs, particularly in chronic diseases, and diabetes always comes to mind when we think of self-administration of drugs in a less invasive way or a less painful way. And there's also an emergence of technological innovations, and microneedles and mechanical arrays included in TD patches were also highlighted in this particular market study, where which have been driving the market as well. Next slide, please. Yeah, so, but there has to be something stopping or delaying progress at least, and that's one of the things we would like to discuss today. So just to launch 3 ideas in the room for discussion. There's definitely a need for better regulatory standards for certain innovations in this field, and microneedles are one of those where we are still kind of waiting for the first microneedle-based product for pharmaceuticals to come to market. And I'm sure Dr. Hedtrich will be able to talk a little bit about this sometimes difficult correlation between in vitro skin models and in vivo results, and also in vivo skin models, which are not as obvious as we might think. For other applications, there's very established animal models, while for skin, it's a bit trickier sometimes. And also the large-scale manufacturing of these devices or carriers or drug carriers. So with that in our minds, I'll just leave it here and open, and I suppose we'll go to the questions.
Thank you so much, Dr. Cordera. I think we can start the discussion right now. Before we start the discussion and before we get to your questions, probably I think it's a good start to define where we stand. So where do we stand in terms of transdermal drug delivery systems? In terms, we're speaking here about clinical translation. Where are we now? What we have succeeded to do, and what are the key challenges? I mean, obviously in general, and we will go into details throughout the discussion. So we can start with Dr. Hedtrich. That's the first on my screen here.
Yeah, thank you very much. It's really my pleasure to be here today. So where do we stand? The thing is that I kind of feel that there has been a lot of investment in academic research. There are many groups doing great research when it comes to skin, how to facilitate intradermal drug delivery. But the problem that I really see is that on the academic side, we've come pretty far, but the translation, there's a huge gap. And so at least from my perspective, I think that when it comes to clinical translation, we are lagging behind, and that as Sarah just pointed out, there are several reasons that account for that. And we have to ask ourselves how we can maybe facilitate that. I think we all know that getting drugs into or through the skin is pretty challenging. Yeah, I really think that we really have a translational problem in that context. That's what I think where we stand.
Thank you so much. Dr. Prausnitz?
Well, I think about the methods of transdermal drug delivery being in 3 kinds of categories. And excuse my voice, I had to give a long lecture yesterday and I'm a little bit hoarse from it. And I think the first category is the drugs that naturally cross the skin well And those are the drugs that we see in the patches today that are making a huge impact. But those drugs are very much limited in number because they have the properties that Sarah referred to before, which only has a few dozen drugs that are good candidates, at least, that have made it through trials. And I'd say then the next category is ones where you then do more dramatic things to enhance the skin's permeability. But many of them are not well targeted to the stratum corneum itself. So there are various chemical enhancers that can have very nice effects on the skin's permeability, but you put too much of it on and then you get side effects. Or you do iontophoresis. You can give some current, but you go the current too high, then you get bad side effects from it. And so I think that has limited this next generation of enhancement methods because you always have this balance of the safety and efficacy, which is inherent in our field. And the safety concerns kick in before the efficacy can get so great that you could deliver, say, macromolecules into the skin. So then I would say there's the third category, which is ones where you have an intervention that is better targeted to the stratum corneum and minimizes side effects that occur deeper down. So you can be more aggressive to the stratum corneum and increase its permeability, but not have the safety or just irritation or pain or other concerns that come with an approach that isn't sufficiently targeted. And so one example of that would be the microneedles that have a big effect on the stratum corneum and have a much lesser effect on living tissue below. But also if you consider ultrasound with cavitation, the cavitation targets the skin surface and breaks it apart. There are certain chemical formulations that are able to target stratum corneum more preferentially. And as they get deeper down in the tissue, they, for example, become more dilute and therefore are not irritating in the skin below. So my bias is that the future of our field, if we want to get into methods that can deliver, especially macromolecules, very water-soluble ones, is methods that can more effectively target stratum corneum and be very aggressive on stratum corneum while sparing the living cells below.
Interesting. Okay, so I will now turn it to Dr. Melero, but let me spin the question also in a positive way, just to touch upon the achievements that we have done so far in the field. So, in your opinion, what is the most promising strategy for local skin delivery at the moment?
Thank you, Heather, because the question before was a little bit difficult to address, but was wonderfully done by Dr. Prausnitz. Actually, I think we, of course, have a lot of challenges to go through, and working on skin is always dealing with reaching the wall, which is the stratum corneum, and it's there. We have to go through it somehow, but I think there are very interesting approaches. Of course, when talking about translation to the clinic, we're talking about big things, because you first have to design the delivery system, then you have to test the toxicity, the efficacy, wherever, in both in vitro, in vivo, and then go to patients. So it's quite a challenging thing to say we are going to translate this, what we are doing on the bench, to the patient. And the more difficult the delivery system you design, the more difficult it will be, of course. But I'm pretty optimistic. I think you know me well, and you always know I'm happy to say positive things. So I really think we are on a good way, at least. It could, of course, always be better, especially in vaccination through microneedles. I think we are pretty far away. There are some systems that are quite straightforward, and I really think they are going to be successful. And for topical delivery, I'm even more optimistic, of course, than for transdermal, because when you let a drug cross the stratum corneum to achieve a local effect, it's always much easier because you need to localize the drug just below the stratum corneum where it will do the therapeutic effect. And you don't really need to reach such high concentrations to reach plasma levels and so on. So In terms of local delivery or to local effects, I'm much more optimistic because, yeah, you don't really need to destroy the stratum corneum in such a manner and continuously for chronic, for example, for chronic treatments. So, yeah, it's my
Okay, thank you so much. I think I will take this point and probably turn it back to Dr. Prausnitz and just ask a question. So, should, in your opinion, should research focus more on local or systemic approaches to drug delivery to or through the skin in the moment? Like, where do you see the sooner clinical translation of products?
I guess I wouldn't say research should focus on one or the other. It needs to be both. If you have a dermatological indication, then the best is to target that delivery to the skin and do it in a localized manner. But sometimes it's a systemic indication. So I would say both are important. The ideal strategy in each case may not be the same. And I think Anna already referred to that. I think one of the distinguishing features of topical delivery is that oftentimes it needs delivery over large areas. So, I mean, if maybe there's just a wart you want to treat, okay, that's in a small area, but maybe you have psoriasis or eczema covering a large area of skin. A lot of the advanced technologies for systemic transdermal delivery are based on interventions that are inherently in small areas. A microneedle patch, applying electricity, applying ultrasound, applying any of the physically based approaches, they don't spread over large areas of the body in a straightforward way. So really, formulation-based approaches are going to be more amenable, I think, to those situations. In other cases where you just need a portal of entry and you want to make the skin very permeable and get enough drug in for systemic effects, then you don't have that constraint and you can be more aggressive with these kinds of device-based or mechanically based or energetically based approaches. So I think that's a distinction that we might think about when considering topical versus systemic.
Okay, interesting. And this is actually a very good transition to Dr. Hedtrich because she is also interested in treating genetic diseases. So this means that the area of application needs to be super big, right? So what's your approach and what's your insights about that?
Yeah, so we started working in that area about almost 4 years ago, 3 to 4 years ago. And so what we are trying to do is to enable gene editing in situ in the skin. And so the neat thing about that is that there is good reason to believe that already correcting only 10% of the diseased cells suffices to alleviate the most severe symptoms of the patients. And so to be honest, we started off, currently we are in the phase, we know how to edit the cells, we have optimized the delivery system, but what you just brought up, how to really make it, how can we bring this to the patient? This is where the problem starts, because currently we work with microneedles, for example, but you can't put a whole human into like a big microneedle. So that's, there's always the question, how will you apply that to the patient? And I think then it comes back to more broadly applicable solutions like ultrasound, what Mark already mentioned, or other things like laser, where you microporate the skin and then you apply the gene editing tools, something like that, sonophoresis, iontophoresis. And to be honest, you don't have to, for those genetic diseases, you don't have to treat the whole body surface. It probably suffices if you treat like half or a quarter of the total body surface. And once this is established, you would preferentially also do that in children, because, those diseases are highly stigmatizing. The patients and their families suffer a lot. And so when you do that in children, you are dealing with a much smaller body surface eventually. And so it gets easier.
Okay, interesting. One thing I want to also touch upon before there is a question in the chat here is Sarah mentioned in her presentation regulatory bodies. So we are talking about research funding. So how this adds to the game or to the challenge of really getting the products that we are working on for years to the market, and whether there are points that we need to consider that are not essentially scientifically oriented when we are designing new projects or developing products that eventually we want to see them in the market. So I will start this time with Dr. Prausnitz. Sorry, I'm really confusing everyone, just based on his recent work on the level of clinical translation.
So I think that creativity is often defined as taking two concepts that people didn't think were related and bringing them together and showing a relationship between them. And so oftentimes where the most creative solutions for, in this case, transdermal drug delivery is when someone thinks, what does a laser have to do with drug delivery? Well, a laser can have something to do with drug delivery, you know, and et cetera. So various ideas come together, but the problem then is that no one has thought about it before. So when you go both to, say, a pharma company and say, hey, pharma company, you know all about drugs, and now we want to use this kind of device or this other kind of technology that you've never thought about before, and let's bring them together because it's going to make drug delivery work really well. And the pharma company is hesitant because this is something very different and they don't know about it. So there's hesitance there. And I think the same applies to the regulatory bodies. You talk to the people there who are very familiar with transdermal patches or topical formulations, and suddenly you say, we're bringing a new technology in. They don't know anything about it. They call up the people in the device part of the regulatory organization, and they don't know anything about drug delivery. And so I think that this is really the heart of the challenge in doing something that's really new, is that the regulations don't exist. And so that means everything gets slower because there's an education process. The regulatory authorities now look to you as the developer to educate them. And I mean, I think they're not resistive and slow because they don't like the technology. They're just slow because they don't know enough about it. And the burden really falls on the innovator to educate the regulators so that they can make a decision, hopefully in your favor, but make a decision based on fact and knowledge. And it just takes time for that education to occur.
Just adding to it, like, how do you think the concept of consumer acceptability or patient acceptability play a role as well in the success of products in the market?
Well, I think it plays a very big role. Although I should say it depends on the nature of the product. So, for example, we've done a fair amount of work on vaccines and think about the vaccines. So, for example, flu vaccine. Flu vaccine is really a consumer product in many ways. It is available and in many countries around the world, very easily available, but you just have to decide to do it. It's not part of a regular visit to the doctor. So if the flu vaccine is something that seems appealing to you and you're not afraid of the needle, for example, and it's convenient for you, then more people will get the flu vaccine. In contrast, if it's the measles vaccine, that's something that's given to children. So now obviously the child has nothing to say about it. The parent probably has something to say about it, but you're in the doctor's office, the doctor says, time for the measles shot, and probably the parent says, okay. And so in that case, it's less of a consumer product and it's more of something that's just part of the medical system. So I think it depends very much on the context in which the drug is being given. Also, likewise, if it's a transdermal formulation that you need to take every day, and it's gooey preparation that you have to rub all over the skin, and it's unpleasant to do it, it doesn't feel nice, and things stick to it, it sticks to your clothing. I mean, there can be bad formulation aspects that will make people less likely to take it. So I do think it can play a very big role.
Okay, interesting. Let me stay on the topic of consumer acceptability, and I will turn it now to Dr. Cordero, just to comment on how this would relate to microneedles, as you're starting to really obviously design projects for transdermal permeation using microneedles. Is this a point that you're considering in your research projects as well to think about patient acceptability of microneedle use?
I think that's fundamental, and it was my experience when I was doing my postdoc. There was a lot of intervention in that area as well. We had, for example, a student in the group who was doing his PhD on antiviral therapy, delivered by microneedles, and one of the parts of his PhD was actually doing focus groups and studying how those devices would be accepted within the community of HIV patients, and asking, and he reported frequently getting questions asked that he, as a researcher in the lab, never thought of, things that people were worried about that he never thought of while manufacturing and characterizing these systems. So I think, as Mark mentioned, it's really, really important, and more important if the product is more of a consumer product than if it's more of a treatment that people will kind of accept. I guess it's a bit of the same kind of dichotomy, I would say, between treatment and vaccines. So people will very easily accept certain level of side effects with a treatment, but they will not very easily accept them with the vaccine. So I think it's kind of the same approach with these systems. So if it's mandatory, if the doctor is telling them to take it, it will not play such an important role, the acceptability. But if it's something that's voluntary, or that it's really going to affect their day-to-day life, then yes, the acceptability is going to be really important. And I may say also in this that there's an organization that the Donnelly lab in Belfast collaborated a lot with in the US called PATH, and they do a lot of these studies with populations and communities that will be the target users of these products, and things come up like wearing patches on your skin in certain countries. Visible patches can be an issue, for cultural and religious reasons and so on. People don't want to expose. So, for example, a question may come from a possible user asking, so is this small enough that I can hide it under my clothes? Or is this, well, can I have a shower with this patch on? Is it going to fall in a couple of days? Very important one. How do I know that it's well applied? How do I know that I've put enough pressure? So I think all of these things are definitely have to be considered while we are developing. And I think that accelerates translation, actually, if we consider them while we are developing.
Okay, that's interesting. Let me go to Dr. Melero, and let me ask a question that was also addressed in the presentation about the role of stratum corneum as a barrier for penetration of drugs. So I don't know if it's generally accepted fact somehow, but maybe could be also overestimated sometimes, that the stratum corneum is the only barrier for penetration or permeation of the drug. So someone can, someone can hear this and say, oh, okay, if the drug really crosses the stratum corneum, then it's just about time that the drug can reach to the circulation. What's your opinion about that, based on your previous work in topical and transdermal delivery?
Thank you, Hagar. The question is not that easy. Yeah, of course, the stratum corneum is always the first barrier, and it's going to be the limiting step. But there are several other barriers. The body is full of barriers. So first is the partition. Every time you change the layer of the skin, the drug has to partition in a different media. And it's always, there are always other people coming into the picture in the skin. You have enzymes, you have macrophages, you have several other barriers that can accelerate the clearance of the drug in the body. But in general, it's usually accepted that if you cross, or the drug can cross the stratum corneum, it will at least reach to a certain point. Then it will be a matter of the delivery system, if it reaches or not, the systemic circulation. But in general, for systemic circulation, you always need to let the delivery system applied onto the skin for at least enough time, of course. Otherwise, the concentration of drug in the plasma will drop. But yeah, there are several other barriers, and they are not that easy to address unless you go to in vivo. And as also Sarah Cordeiro mentioned, in vivo models for skin delivery are not that easily translated to human. I'm working a lot with pharmacological models now for different kind of inflammation diseases, because none of them is really a good, is not really good mimicking good the skin that you have in a human. And yeah, all the models have some positive aspects that you can rely on, but several or many more problems. So once you address one question, then you come to another. Was my model appropriate or not really? Because you generally develop the inflammation through chemical hazards to the animal. So sometimes it's a critical point if you apply the delivery system right before, afterwards, because sometimes your own delivery system is preventing the chemical to develop the condition. So it's really a complex question. But yeah, okay. I think we still have a lot of work to do on that.
Okay, so let me go a step backward, just so that everyone is on the same page. So before talking about what would be a good model to evaluate transdermal drug delivery systems, probably it's important to also discuss with others about, one sec. About what other challenges or barriers again, is transdermal drug permeation. Like here we are really focusing on transdermal, not topical. Transdermal permeation other than stratum corneum. So maybe Dr. Prausnitz can start.
So I would like to mention something that is, I think is important. It's not a permeation barrier, but there are a variety of drugs that actually can get across the skin effectively, but the drug itself irritates the skin. And so then that drug is simply not a candidate for transdermal delivery. So that's one example where we do need to think about the cells that are living below the stratum corneum, because that could be a reason why a drug is not a candidate. But beyond that, let's say you use some method to make stratum corneum very permeable. There are the next layers. And the viable epidermis, and there's a basement membrane between the viable epidermis and dermis, that can now become a barrier. There's some debate in the community about the existence of tight junctions within the viable epidermis and the role that they might play in limiting transport. I think empirically we do know that as you start using large molecules, the viable epidermis does become a barrier. And in the past, we haven't worried about it so much because the stratum corneum is such a big barrier that, oh well, if something gets across the stratum corneum, whatever is below will not be a barrier. But now we have the situation where it becomes an issue, again, especially for macromolecules in epidermis.
Okay, interesting. So I will go now to Dr. Hedtrich to talk about like what other barriers that you can think of, and also probably start discussing what would be a good model, since this is also a big part of your research work.
Yes, that's one of my favorite questions, actually. But starting with the barriers, maybe I think we should also keep in mind the adipose tissue, basically, which is in the subcutis, because lipophilic substances may, it's a deep compartment, and lipophilic substances may accumulate there, and then this might be another barrier to transdermal delivery, for example. Yeah, and with regard to the models, so the thing is that, and I think this is really not only a problem in skin research. We in general there's a model dilemma in biomedical research, and so the thing is that very often animal models are poorly predictive for the human situation. And when it comes to skin, there are so many striking differences. I mean, still mice are most frequently used, but just look at the anatomy of a histological section of mouse skin. It has on average 2 to 3 epidermal layers. Human skin has 18 to 20 epidermal layers. So getting drugs into or through mouse skin is so much easier than doing that in human skin. And there are so many publications out there, also in very high ranked journals, that have some mouse models, and then they claim, okay, we can deliver XYZ also with gene editing tools, for example. Very high ranked journal, and show that in mice. And this literally tells you zero about what's going to happen in humans. And so this is just something what Anna already mentioned, that we really have to choose our models carefully. And there's a very nice saying that I quote very often, which is from a British statistician who said, once all models are wrong, but some are useful. And so I honestly believe rodent models, except when it comes to toxicological testing, are not well suited for skin research. That's my personal opinion. There's also, besides the anatomy, there are mice and humans, so the genome, there's only a 30% overlap, for example. And also when it comes to emulating human diseases, we do a lot of research in atopic dermatitis. And the thing is that this is a disease that is not spontaneously developed by most lab mice. So what happens is you artificially induce a phenotype that somehow looks like eczema, and then you start interpreting that and use that to study the pathogenesis and to test drugs, and then you extrapolate that to humans. And this is where the problem starts. So what I really think what we need is, when it really depends on the context. So when we want to test the delivery efficiency of drugs into human skin, what really is an excellent starting point is working with excised human skin. Freshly excised human skin, this maintains the physical barrier. I know there are some ethical problems depending also on the country. It may be easier or more difficult to get access to that, but I think this should be really the gold standard. And then, I mean, I'm a big fan of reconstructed tissue. So we do a lot of bioengineering trying to emulate the disease phenotypes as well. And this also has its limitation because it's not in a living organism, but we know now, we are aware, and there's a large body of evidence showing that just studying it in a living organism doesn't hold the solution because the predictive value remains low. Yeah.
Okay, that's I personally agree with many of this, because this is also a focus of my research group. But let me go back to Dr. Prausnitz, because this is really an important point. Like, I mean, there is one saying that the best model for humans is actually a human patient. So that's the best model. So what's your say on that?
Well, I mean, of course, I think everyone will agree with the statement you just made. But of course, we can't use the human all the time. So what can we do that to as a compromise? And I think the choice of model really depends on the scientific question you're asking. So if your objective is to study the mechanism by which you can increase the stratum corneum permeability, then cadaver skin is okay, because the stratum corneum is not terribly changed upon death. And human cadaver is ideal, but maybe then we do a lot with pig skin from a slaughterhouse, and that's fine. But if you care about living cells, if you want to do, I mean, especially if it's topical delivery and you want to do something to the living cells, now dead skin is just obviously out. So you can go to human skin equivalent, cell culture-based, tissue culture-based skin, and there the stratum corneum barrier may not be quite as good, but it can be pretty good. And now you can study actual physiological processes in the skin, or even better is surgical samples from human surgery, as Sarah just mentioned. A complication now comes that a lot of skin disease has a strong immunological component. So now, if you excise skin, you can, the immune system is partially present, but is not fully present because you don't have the rest of the organism. And so you have to then accept that limitation. So I think it's, you do the best you can based on a good understanding of what your scientific question is and what mechanism is involved, and then make sure the elements that you believe are relevant to that mechanism are present. And you know, that's then going to depend how hard you need to work to recapitulate the whole human, or can you just have some smaller part of the challenge present, because that's the part you care about.
Yeah, that's for sure. Okay, so let's remain on the same topic and now ask a question from the audience. So Marwa had a question to, probably I can ask Dr. Cordera to answer this. Do you think human skin equivalents are better for predicting translational value?
Well, if you don't mind, Hagar, I'll totally pass the ball on to Sarah.
Okay.
Because I think she'll have a bit more information on this. And I think, sorry for just stepping in, but I think that question is related as well with the following one, which talks about developing a living model of human skin with 3D bioprinting and
okay,
culture.
So maybe Sarah can then answer both questions about the equivalent models as well as the living model of human skin using 3D bioprinting and culture, and maybe including the organ and chip models as well.
Yeah. So I really think, and this is, I would like to second what Mark just said. So I think it has human skin and human skin equivalents do have the better predict translational value or higher predictive translational value, depending on the scientific context. So if it's the right scientific context, then yes, I think it is more predictive. I'm 100% sure that it's more predictive when it comes to skin absorption. Although, as Mark also mentioned, skin models have a weaker stratum corneum, but also from our studies and many other labs, they are still very restrictive when it comes to nanoparticle penetration and things like that. So in that sense, they recapitulate human skin barrier very well. I think when it really comes to testing the delivery across the human skin barrier, I'm a big fan of those freshly excised surgical tissue. So you collect them from the surgeon, they're still warm, you take them to the lab and you can keep them alive for up to 48 hours. Some people claim they keep them up to 96 hours, but we always stop after 48 hours. At some point it just gets smelly and I'm not sure if the tissue is still viable anymore. So I personally think that bioengineering human tissues is really the future for drug testing in general, not only for skin. So I would like to answer that question with the yes, that's the future. And then developing a living model of human skin, yes, this has been done frequently. So that's, and skin models are actually probably the most advanced tissue models that are currently available. Bioprinting, I'm not a huge fan of bioprinting because the thing is that these are usually not very mature models or not well differentiated models, although they have some advantages when it comes to positioning the cells accurately and generating models of high cell diversity. But then the cells are just sitting there and they are not mature. They haven't differentiated yet. So this is a process that would have to come after the bioprinting. And very often you see publications where they bioprint the tissue and then say it's ready to use. But it's mostly not. So I'm not sure about that. I think it's an exciting technology, but we'll have to see where this goes.
Okay, interesting. I like this answer so much as well, but also just for personal biases. But let me go to Anna and ask a question that is very a hot topic right now. The effect of sex and gender. At least here, I mean, in Canada, like whenever you submit any projects, you need to really address how you are going to address the effect of sex and gender. So this is one of the questions from the audience. So if you can start the discussion here.
Yeah, thank you. I think it's a really interesting topic. Here in Spain, it's also the way we are starting to, in all the proposals that we have, we need to include the sex and gender question also. So I'm not really working that much on this on skin, although what I wrote in my last proposal was that the delivery systems I wanted to develop were they were to localize they were designed to localize the drug onto the skin and not systemically, and that could very much help some women that for example, would like to get pregnant, but they cannot skip their systemic treatments for psoriasis or dermatitis, which is the topics I want to address. And delivering the drug through the topical route and localize it on the drug and not to provide a systemic effect that could help them to decide whether to have children or not, for example, or also to, even if they continue with the treatment, to do it in a safer manner, or to at least to believe the treatment was going to be safe. But it's really a great topic. And in other works I'm doing in, for example, well, with a colleague that I work with at the University of Valencia, we are working on nose-to-brain delivery of certain drugs. And we have designed it accordingly to a gender design. So we administer it to a male Wistar rat, to a female Wistar rat, and we do observe differences in their behavior. We are working on pain, and on the pain which is affected by alcoholic dependence. And it's really a huge difference. So I really think maybe in the skin, I couldn't see till now. Maybe my colleagues can do and can comment further on this. I didn't think of anything else, any difference, huge difference we could have apart from the hair follicle number and so on. That could, of course, be a difference, that's for sure. But I couldn't think on that way. But as I tell you, in the behavior experiments, we really have very different pharmacological effects, whether it's female or male. So probably with the skin, we will also see this.
Okay, so I will go to then Dr. Prausnitz, as Dr. Melero recommended. So like, do you have additional insights on this topic, or maybe other factors that you think are really important to consider when we are thinking about clinical translation of transdermal drug delivery systems?
Well, I think we've touched on some of the topics. One of them is in terms of the acceptance. Well, we talked about the patient, but I would take it even more broadly, the healthcare provider as well, and the healthcare system as a whole. And I think the field of drug delivery and pharmaceutics inherently thinks about that, I think, more than maybe some others do. And so I think that is very important for that success. We also talked about the regulatory and the barriers that exist there, and being able to educate. But I think something that we haven't talked as much about is that in order for people in drug delivery to be successful, usually we need pharma to buy in, because usually the driver in terms of the money to pay for it, as well as just the infrastructure to implement it, comes from pharmaceutical companies. And I think that they are inherently conservative, and maybe appropriately so, because our field is, the field of pharmaceuticals is just littered with failures, right? For every drug that makes it, there's been so many that have failed along the way. And so I think that is a real barrier to the translation, where our community, the pharmaceutical community as a whole, is so risk adverse that we need to demonstrate, one, reduced risk, but also real increased value. If the value is a little bit better, it's probably not going to work, because the risk is so high to make any kind of change that I think that puts a big burden on us to demonstrate the great value that can come that will justify the risk that pharma needs to take.
Okay, interesting. There is a related question from the audience, and so Mohammed is asking, do you think if a new technology is translated for cosmetic purposes, would it help boost regulatory aspects for this technology for therapeutic purposes? And the first thing that comes to my mind is the dermal roller. So we use the dermal roller all the time, and still there is a big challenge in translating microneedle patches. So what do you think about that, Dr. Prausnitz?
Well, I do think that the rise in using microneedles for cosmetic purposes is generally helpful for advancing the technology for pharmaceutical purposes as well. The dermal roller and related technologies were really underneath the radar for the FDA, and they were being sold. And then the FDA came out with a formal position statement opining that if the derma roller or such devices go past stratum corneum, then they become medical devices. And so that then has had perhaps a good effect in that FDA is now thinking about these systems, but a bad effect in the sense that they took an extremely conservative point of view. Anything that goes past stratum corneum, well, if it doesn't go past stratum corneum, it's not a functional microneedle. It doesn't do anything for you. So basically, they've completely thrown the field over into medical device very clearly. And clarity is always good from a regulatory standpoint, but it does create now a greater burden that if anyone's thinking about doing any kind of a microneedle-based approach, it seems like FDA is going to say that's a device, and so that's going to add some regulatory burden because now you've definitely got a drug device combination.
Okay. That's interesting and challenging. I have another question to Dr. Hedtrich on the same topic, probably. So Dr. Prausnitz mentioned about taking risk and value, so balancing value and risk in our decisions. So how do you judge the risk and value of commercializing a potential product from bench to the market? Like,
yeah.
Do you have insights on that?
Yeah, I can give you a very concrete example from my own experience. So my lab has been working on a topical substitution therapy for genodermatosis, so rare genetic skin diseases, for almost 10 years. And so we, together with a collaborator who is a polymer chemist, we came up with some thermoresponsive nanogels that beautifully and highly efficiently delivered large molecules into the skin. We were able to show that this resulted in the recovery of the skin barrier function. Then we wanted to take a step further and to spin that out into a company and things like that. And we talked to a couple of pharma companies and potential investors. And the thing that we didn't think about is that those polymeric nanogels, so they were all very risk averse because these are new chemical entities and they would have to run a complete tox on those polymeric nanogels. And this costs about roughly $3 to $4 million. So this is very high risk. And this basically tanked the whole story for us. So we went back and we pivoted now, and we are, for the gene editing project, for example, we are working with lipid nanoparticles. And I mean, they have been injected into billions of people worldwide. So there is a completely different level of acceptance already. They are already approved. They are considered, they have the GRAS status. And so there currently we are also in negotiation with some potential investors. And the thing is, this is a completely different story. And so my suggestion would be, or my advice would be, if you really would like, or if you think about translating your approach, think about how to really generate a lean product or a lean approach, because the more complicated it gets, then investors, pharma companies, they won't invest in that, and they won't take it a step further because it's too high risk, as Mark also pointed out.
Okay, good. So we do have like 1 minute, but I would probably ask all the speakers to, so we are all researchers, to think about or to give us your insights about what are the big questions that we as a society need to answer in the next period in order to move more transdermal drug delivery system to the market. So I will start with Dr. Cordera.
Okay, and I will bridge from what Sarah just said, because it's something that resonated and resonates with me since the CRS annual meeting in New York 2018, where Professor Jain, in a session for the YSC, he said, we're developing very fancy nanoparticles with lots of functionalities, targeting, and that applies also to transdermal delivery. And we're developing these very fancy nanocarriers that no one will be able to pay for. So not companies, not healthcare systems, not patients, not insurance companies, if we're talking about private healthcare systems. So that's something that resonates in my head all the time, and it's something that I try to keep in mind in my research lines, is to stay simple and stay and think of of a way that we can develop new therapies or new approaches to drug delivery that are not going to be unaffordable, so that they can actually make an impact in patients. So that's something that I would leave as a message. Stay with me.
Very important message. Dr. Melero.
Thank you. So I really fully agree with Sarah. I'm in the same way. So the simpler, the better. I don't know, it's also a question of funding in our country, in our research lab. The easiest that you can keep it, the better, because you always face a lot of more challenges. Even though you think on paper when you draft a proposal, it's simple, then all the problems will arrive afterwards. So if you face the problems, then it will get worse when scaling up, when translating, when the patient has to apply it. So they have to be simple from the conception to the application. Also, the patient needs to, as it's already been pointed out, it should be easy to use. Like, I really think of the sentence you said, Sarah, about how do I know if I applied enough pressure, things like that. So yeah, I agree. It's always easier to convince with a simple manner than to complicate it too much.
Okay. Thank you. Other questions do you think that are important to address, Dr. Hedtrich?
Not really. I would like to second what Sarah and Anna already said, but I think it's really important to think to choose the model wisely, because a project stands and falls with the right model.
Okay, interesting. Dr. Prausnitz?
Well, I will agree with what has been said. We have crazy ideas that we like to follow, but at the end of the day, it's the simpler ones to implement that will have an impact. But at the same time, I just want to layer onto that, not to be incremental. So and that's fine if you are in product development and you need the next generation of something for your company so that you can get a new patent, or you can get some advance. Even incremental changes can be big. But if you're in the area of advanced drug delivery research, whether in academia or at a company, assuming your company will allow you to think big, I think the biggest challenge is to really think big, to have ideas that really change things in a big way, but at the same time, allow a straightforward implementation. And that, I think, is the ultimate challenge, to combine those two things.
I think that's a very positive note to end with. Thank you so much for all the speakers for your time today. I hope I have covered all the questions in the chat, and I do apologize if I couldn't or I missed any question. If so, I mean, you still have the opportunity to meet the speakers in the annual conference that is coming soon in July. So hopefully we welcome everyone there, of you in Canada. So again, thank you everyone for today, and we hope we meet for the following events, probably after the annual meeting, and thank you for today.
Thanks, everyone. Thanks, Hagar.
Thank you. Bye.
Thank you.
Bye.
Bye.
Bye, everyone.